like few topics which can come for 3 marks question. We will also see those because we are going in order, right? So what is biochemistry? The chemical reactions which are taking in the human body, human cell, okay, which is basically composed of various metabolisms, like whatever we eat, carbohydrates, proteins and fats, how they are metabolized in our body and how it is converted to energy like ATP, and so many other things like vitamins, minerals and all we usually study under this subject biochemistry. Okay, bio is life, the chemical reactions what are taking place in a human being is generally studied under biochemistry. Okay, so basically we have two types of cells, no, like prokaryotic and eukaryotic cell. Prokaryotic cell is nothing but the bacteria and all. Eukaryotes are like the well modified human cells. So this eukaryotic cell structure, just learn this structure better. Sometimes you may get a small question like, you have been studying right from your schooling, like any like eukaryotic cell, it has a cell membrane, nucleus, nucleoplasm, nucleolus, Golgi complex, ribosomes, endoplasmic reticulum, rough and smooth endoplasmic reticulum, lysosomes and all, isn't it? Now when you talk about
Yes.
mitochondria, we know it, these are the powerhouses of cells, isn't it? So then
Yes.
ribosomes, they are factories which are concerned with the production of proteins. Endoplasmic reticulum also gives support to that ribosomes only, which also facilitate protein synthesis. Peroxisomes will protect the cells against any harmful substance like antioxidants. Okay, then lysosomes, suppose if the lifespan of a cell is over, suppose if RBC, what is the lifespan? 120 days. So after which you will see certain chemical substances are released inside the cell, enzymes which are autolysis. That means that will cause autophagy, auto digestion. So lysosomes are suicidal bags which will
Yes, ma'am.
kill the cell after the lifespan of that particular cell is over. Okay, so any of these organelle can come for 3 marks question. Okay. Mm,
Yes.
all is just go through better. They are not so like tough.
Yeah, ma'am. Yeah, ma'am, it's not. Actually, ma'am, in the biochemistry book I have, all this is not given. I mean, simply like this, all the peroxisomes and all of that. They directly started with one thing.
Acha, acha.
They started with the structure of cell, ma'am. The
This
first
is the
topic.
same thing, structure of cell only, Anjali.
Yes, ma'am. Yes, ma'am.
Ah,
They directly went off to fluid
ah.
mosaicism
Carbohydrates,
and then
no.
myelin,
Yeah, this
myelin.
will take only few minutes. I thought I will not leave anything. I'm going to
No,
start
no,
topic.
I'm just saying that in this book it's given more clearly, I think, more than this book.
Mhm. Whichever book you feel comfortable and convenient and easy to learn, you can follow. There is nothing like you have to follow Satyanarayana only. No, Vasudevan is also equally good. If you are comfortable with Vasudevan, you can continue with that book as well. Check whichever is easy. Okay, so now see, sometimes you get a question like apoptosis. Apoptosis is programmed
Yes.
cell death or cell suicide. So that means once the lifespan is over, you will see apoptosis details you will be learning in second year, in fact, in pathology. Okay, so that is only a small introduction what we have. You can just go through this summary once, okay, before you start the chapter carbohydrates. Okay, now, so what are carbohydrates? Basically, the food what we eat, it is made up of macro nutrients and micronutrients. So macronutrients are the things what we consume in larger quantities. Micronutrients are the nutrients which we consume in milligrams. Like for example, macronutrients, we have carbohydrates, proteins and fats. We consume them in grams, whereas micronutrients we consume them only in milligrams, like vitamins and minerals. Okay, both are equally important for our health. In order to produce energy, generate energy and to sustain our life, we require macronutrients. Okay, that means carbohydrates we require, proteins and fats we require. And to like there are certain chemical reactions which are taking place in our body only in the presence of certain micronutrients. That means iron, calcium, these are minerals, and vitamins like vitamin A, D, E, K, B complex and C. All these vitamins are also, though they are required only in milligrams, but deficiency of such micronutrients also will result in diseases. That means micronutrients and macronutrients both are equally important for the maintenance of our life. Okay, so now coming to the definition of carbohydrates. See, basically carbohydrate, like when you see, hope you know this, the formula of glucose, C6H12O6, right? Isn't it? So
Yes, ma'am.
that means carbohydrate is made up of carbon, hydrogen, and oxygen, isn't it? So carbohydrates are defined as polyhydroxyaldehydes or ketones or compounds which produce them on hydrolysis. Okay, they are polyhydroxyaldehydes or ketones. We'll see the structure, then you will remember more. So now basic functions of carbohydrates, like whatever we eat, like rice is, most of the rice is carbohydrate only. Potato is a carbohydrate. So many things are there. Sweet potato is a carbohydrate. Many food things what we consume in our daily diet, it is like even wheat has a lot of carbohydrate. That means what? This is the most abundant dietary source of energy for all living organisms. So carbohydrates are precursors for many organic compounds. That means from carbohydrates, proteins like amino acids and fats also can be synthesized. Okay. Then carbohydrates is also going in the formation of cell membrane structure. Okay, it will have like lipoprotein and glycoprotein structures are there in the cell membrane where you will see carbohydrate is contributing. It is also structural component of many organisms. Okay, like cellulose. See, cellulose we will be studying like at the end of this chapter also. See, cellulose is something like fiber which is actually not digested in our body because we do not have like cellulase enzyme which is actually responsible for the digestion of cellulose. Okay, the roughage fiber what is there, it's very important. Though it is not digested, it will add bulk to what we eat. It gives fullness, yeah, we ate enough, we will get that feeling. And it also prevents constipation. Okay, and in long run, fiber also protects us from colon cancer. So fiber is very important. For like about fiber only you will get every year question for 3 marks or
Yes.
5 marks. So like that fiber is also basically made up of
I'm
carb.
like, I mean, fiber. What is the importance of fiber?
See, fiber is actually not digested
No,
in
no,
the
I'm
body.
asking the question that comes. Is it in? Will they ask you about
No,
the
they will
importance
just
of
give one word question for 3 marks or 5 marks. Question will be dietary fiber. That's it. Dietary fiber. It can come either in physiology or it can come in biochemistry as well. There also we have a system called digestive system in physiology. So in both the question papers, this is most popular question, dietary fiber. Okay. So, and carbohydrates also serve as storage form of energy. That is, if we are eating rice or chapati made up of wheat in excess, what happens? The excess carbohydrate is stored in the liver in the form of glycogen. We know basically
yes?
carbohydrate is like it's a glucose, like where we have polysaccharide chain of glucose. That means it will be either glycogen or it will be in the form of starch what we eat. So suppose if we have taken it in excess, whatever is required for the body will be utilized. Okay, and the remaining part which is not utilized by the body will be stored in the form of glycogen. Especially you will see the storage will be in liver and also muscle. Liver glycogen and muscle glycogen. Okay. Now coming to classification of carbohydrates. So we have basically carbohydrates classified as three major groups like monosaccharides, oligosaccharides and polysaccharides. If you find only one sugar unit, you call it as monosaccharide. If you find 2 to 10, below 10 sugar units, we call it as oligosaccharides. If sugar units are more than 10, we call them as polysaccharides. Okay. Now, monosaccharides. I said monosaccharides are nothing but they are simplest form of carbohydrate. Simple sugars. They have only one sugar unit. Okay, which cannot be further hydrolyzed. Okay, so monosaccharides are further divided based on the functional group. See, the monosaccharides are any carbohydrate, they may have functional group either aldehyde or they may have functional group ketone. If they have aldehyde functional group, that is CHO, we name them as aldose sugar. Aldehyde sugar is aldose sugar.
Not sure.
And if you have the functional group as ketone, we name it as ketose sugar. Okay, so glucose is aldohexose, that means it is having the functional group CHO, aldehyde. Okay, while fructose is ketohexose, both will have same structural formula, both will have C6H12O6. Glucose is having functional group aldehyde, and ketone, like fructose has functional group ketone. Okay, and when you just check the sweetness of glucose and fructose, fructose is more sweeter when you compare to glucose. Okay, so now let us see, you got an idea what is monosaccharide. Okay,
Yes, ma'am.
now let us see the classification of monosaccharide with selected examples. This you have already learned now, isn't it? Hexoses, that means if you have 6 carbon units, we know the formula 6 C6H12O6, that is like hexose sugar. So it can have an aldehyde functional group CHO, then it is named as glucose, and it can have a functional group CO. Just now we have seen, isn't it, C double bond O, like fructose. Similarly, we may have 3 carbon unit called as trioses. Okay, structural formula will be same whether it is glyceraldehyde or dihydroxyacetone. One is having aldehyde, the other is having ketone. Similarly, you just completely learn this table. Okay, monosaccharides, aldoses, ketoses. So that is given, and I hope you understood this.
Yes, ma'am.
Oligosaccharides, I said the sugar units are ranging from 2 to 10, isn't it? That means the monosaccharide molecules are 2 to 10 in number. Then we name it as oligosaccharides, which are
Ma'am, so
further subdivided
polysaccharides for
more than 10.
polysaccharides.
Polysaccharides are more than 10 sugar units, more than 10 monosaccharides. You call them as polysaccharides. Okay. So if it is less than 10, we name it as,
Uh, pol. Ma'am, if it's more than
if it is less than 10
less than
monosaccharides,
10 oligo.
oligo. More than 10,
Uh, polysaccharide.
polysaccharides. Yes, very good. So under oligosaccharides, if two sugar units are there, we call them as disaccharides. Do you remember any example of disaccharide? Two sugar,
Uh,
two glucose.
ma'am, lactose.
Very good. Yes, very good. Do you remember any polysaccharide? Any sugar units more than 10?
Ma'am, glue.
Glucose is only one sugar unit, no?
Right. Oh,
Monosaccharide, that is.
I mean,
Today you are sleepy, Anjali.
no, ma'am, inulin, ma'am.
Huh?
Can you
Inulin,
take
inulin,
inulin?
right?
Yes.
You told inulin, right? Yeah.
I wanted to say inulin, so I got confused. Glucose. Yeah,
Inulin is
inulin.
also nothing but starch only. You can use that. Okay, so that means that is a polysaccharide which has more than 10 sugar units. Okay, so like that, see, we have again homopolysaccharides and heteropolysaccharides. Now, what do you mean by homo? If it is made up of all glucose units,
Yes,
a
ma'am.
like you have a chain, polysaccharide chain, which is having more than 10 monosaccharide units, which are all same, all 15 glucoses, 20 glucoses, 100 glucoses, so many, all glucoses if you find, then you call such polysaccharide as homo. Best example is starch and glycogen. Starch is found in plants and glycogen is found in animals. Both are homopolysaccharides only. Both are exclusively made up of glucose only. Understanding? Then heteropolysaccharides, in the sense it will not have same monosaccharide units. One glucose may be there, one fructose may be there, something like that. It will change. Not uniformly only one monosaccharide, then you call it as heteropolysaccharides. Under heteropolysaccharides, in this chapter, we are going to learn about glycosaminoglycans, which is 5 marks question. Let us go in order so that we will focus on important things. Now, monosaccharide structural aspects, like we have stereoisomerism. So that means like same structural formula they will have, but when they are allowed to dissolve in a solution, they will show a specific type of rotation, either clockwise or anti-clockwise. That's what is spatial configuration. So which will differ from sugar to sugar. So that is important characteristic of a monosaccharide, like glucose and fructose. So they are compounds, they have same structural formula. Best example, glucose and fructose. Both are monosaccharides, both have same formula. One is aldose sugar, the other is ketose sugar. Because their functional groups are different, when they are allowed to just dissolve in some water and when they are exposed to some radiation, they will start showing a particular type of rotation, which can be either clockwise or anti-clockwise. So that is, they differ in their spatial configuration. Okay. So this also we can go through better. Monosaccharides.
Yes.
Already you have
Try.
learned triosis, tetrosis, pentosis from the other table, isn't it? Here, you
Yeah,
already
ma'am.
know these
Yeah,
names.
ma'am.
Huh? So once you learn this, a little more points have been added. Like triosis, when you talk about glyceraldehyde, where is it situated? It is found in the cell size phosphate. What is the importance of glyceraldehyde? It's an intermediate in glycolysis. When we do carbohydrate metabolism, we will come across many cycles like glycolysis, gluconeogenesis, HMP shunt, and all. Okay, so during which you will come across this substrate like glyceraldehyde. Okay, that means it is taking part in glycolysis. You can write like that. Even dihydroxyacetone, it also takes part in glycolysis. Like that, you have many examples. Among these, because time is there, I'm asking you to learn many things. Okay, but otherwise, the most important things here are sucrose, lactose, and maltose you read thoroughly. Glucose and
Yes.
fructose you read thoroughly. In whole of this table, you can make a note.
Yes,
Okay,
ma'am.
ribose is also you can make a note. Because sucrose, lactose, maltose, glucose, fructose, they can come for 3 marks question individually.
Yes,
Okay.
ma'am.
Now, fructose, glucose already we have studied, fruit sugar and all. Isn't it? Which is sweeter, I said? Is glucose sweeter or fructose sweeter?
Ma'am, fructose.
Fructose, fruit sugar, isn't it? Okay. Yeah.
Yes, ma'am.
Huh. Now, see here, sucrose, 1 minute, beta. So, these examples you need to do in detail, beta. Like sucrose, very simple.
Yeah,
We
ma'am.
find in cane sugar and it is most commonly used in
Ma'am, so like
table.
glucose and galactose, should we like know that also? Like what is
Yeah,
constituting?
galactose also, yeah, milk sugar, yeah, lactose. Most important ones are glucose, fructose, sucrose, lactose, maltose, beta. Other than that, also, if you can remember this table, that is well and good only. Because if any bouncer comes, also, you will be able to write something, isn't it?
Yes.
Just go through CR question bank you have, right?
Yes, ma'am.
Check what questions have been asked from this chapter.
Yes, ma'am.
Hmm. In here I'll be telling, but still you also go through
Yeah, ma'am.
once. Okay. Now see, so there are something like D and L isomers. So that means what? So see here, this is the structure of glucose. So which is showing some dextro rotation. Suppose if it is clockwise, there is one more structure of glucose. Only configuration has differed on fifth carbon atom. H is on left side and OH is on right side for D form. OH is on left side and H is on right side on in L form. So such slight differences in this structure, they are generally considered as isomers. So D and L isomers are mirror images of each other, as you can see here. The spatial orientation of hydrogen and hydroxyl groups of fifth carbon, this is adjacent to terminal primary alcohol. This is only different. That's what is written here. Okay, if you just refer and read this, you will understand.
Yes.
Okay. So optical activity, I was just talking about, it will rotate if it is allowed to dissolve in a solution like sugar. You dissolve in water, and when you expose it to some radiation, what happens? It will start rotating either in clockwise or anti-clockwise. Okay, so this is optical activity, beta. Dextro-rotatory and levo-rotatory, like based on whether it is rotating in clockwise or anti-clockwise. So just go through. These are actually important for practicals only. Theory-wise, we don't get much questions. Okay.
Yes, ma'am.
So what is racemic mixture? This is also again actually important in practicals, like D and L isomers are present in
Yes.
equal concentration. Okay, you call
Yes.
it as racemic mixture. So they will cancel each other, hence there will be no optical activity in case of racemic mixtures. Okay, Orestol or epimers are important, beta. So for epimers, first let us see the pictures, then you will understand. Epimers, enantiomers. See beta, epimers, like picture is given 2.4, let me check that, and then it becomes easy also for you to understand. Yeah. See, this is structure of epimers. Now, if you observe this, you are finding this is the structure of galactose, glucose, mannose. If you just compare this glucose and galactose, both of them are having how many carbons? 6 carbons only. Okay. Now, if you just observe the fourth carbon, so these are quite oppositely placed. Rest
Yeah.
all look same only. This is D-galactose and this is D-glucose. So the configuration is different only on fourth carbon. When you take D-galactose and D-glucose as examples, they have the same structural formula, but they differ the configuration of OH and H across fourth carbon. Now, when you just take these two as comparison, what is different, Anjali? Which carbon?
Ma'am, the second
Very good, loudly.
second one.
Second carbon. So second you can see hydrogen OH is placed for glucose on the left and right side, and it is vice versa for the mannose. So they are epimers with respect to second carbon, isn't it? D
Yes, ma'am.
glucose, D mannose are epimers for second carbon atom, whereas D galactose and D glucose are epimers with respect to fourth carbon. Okay, now let us read the definition. You will easily understand because epimers can come as a 3 marks question again.
Okay.
See, if you take two monosaccharides which are differing from each other in their configuration around a single specific carbon atom, they are called as epimers. So
Ma'am, so
glucose,
it should be just one carbon.
yeah,
I mean, it can any position.
any position. So glucose and galactose, one carbon. Which carbon they are differing? Fourth carbon.
Second one. Uh, Glucose and
See, glucose,
mannose
galactose,
again.
fourth carbon.
Galactose, that is fourth. Yes.
Fourth.
Glucose and
Glucose and
mannose,
mannose,
second one.
very
Second
good.
carbon.
Same thing is written there, beta. If you understand concept, you can write in your own words. Okay. Same thing is okay. So glucose and galactose they are with fourth carbon, whereas glucose and mannose they are epimers with regard to second carbon. So there can be a change interaction also. Suppose if the enzyme like epimerase is acting, glucose can be converted to galactose. Galactose can be converted to the mannose, what we have seen just now, like this. Glucose can be converted to mannose, mannose can be converted to glucose, galactose to glucose, glucose to galactose. What is the enzyme? Epimerase.
Epiphany.
Okay. So that is epimers. Sometimes a question for 3 marks can come. What are epimers? Now coming to enantiomers. Again we will see the picture first, 2.5. Then you will understand better. See here, L-glucose, D-glucose. See this is all glucose molecule only. One is levorotatory and the other is dextrorotatory. If you just observe here, how do they look? This side you have oxygen, like which group
Yes,
is
ma'am.
this? Aldehyde group, no? But the other
Yes.
side, they look like mirror images, isn't it, Anjali?
Yes, ma'am.
Hmm? So this, oh, you just write whatever is written here, this side. Right side things will be written on left, left side things will be written on right for L-glucose and D-glucose. So you call them as enantiomers. Okay. These are special type of stereoisomers that are mirror images of each other. The two members are designated as D sugar, D glucose, L glucose, which is sano, right? Majority of the sugars in higher animals, including man, we come under the category of dextrorotatory, D glucose. Okay. Then,
Yes, ma'am.
so suppose if there is no mirror image, then we use a different word called as diastereomers. Okay. So where stereo isomers that are not mirror images of each other. Okay. Diastereomers. So that is about epimers and enantiomers and diastereomers. Structure of glucose actually not required, but anyhow for the sake of epimers and enantiomers and many other things you will be learning. So automatically you will be learning, but as such structure of glucose nobody is going to ask. Okay.
Yes, ma'am.
So then mutarotation is important better. So what is this mutarotation? Basically, let us see the picture again. It becomes easy for us, mutarotation. Just check this, I'll tell you. See here, muta-rotation of glucose. You are finding this is D, dextro-rotatory type of glucose beta, which is alpha. Then this is D glucose. You are finding this is in the active form of rotation, whereas this is just at resting stage. That means it is not dissolved in any solution. Okay, it is at rest. There is one more different type of rotation you are finding with glucose, which is beta. Okay,
Beta.
so in alpha and beta, different forms. What are we finding? See here, this is same like enantiomer just now we saw mirror images like different. See this side H H H O H. So in the place of this side, this side things have come this side. Okay, just try to observe the formula is same. This is also glucose. This is also glucose. This is also glucose. But configuration is differing, and you have to practice this for sure.
Yes.
Okay, so let us see this. You have this in mind. One is like optical rotation values are given. For alpha D
Yes.
glucose, how much it is?
One 112.2
2 degrees. Okay. For
and
beta D glucose, how much?
18, 18.
Yeah, 18.7. Now let us read like what is this muta rotation? Just a minute. Read this once. Muta rotation.
Uh, Ma'am, out loud.
So hope you have gone through this mutarotation definition.
So, so the alpha anomer, which is 112.2, it attains an equilibrium with a constant value of 52.7,
Hmm.
and then in the presence of an alkali, it further decreases to a value of 18.7.
Yeah,
Then it becomes the beta glucose.
nice, very good. Yeah. Okay, so that is mutarotation. Change in a specific optical rotation representing interconversion
Represent-
of alpha and beta forms. Nice, very good. So this is the only question which is usually asked, Anjali. Mutarotation is very important question which can come either for 3 marks or 5 marks. Okay, beta? Then rest all are like 2, 3 markers only. Only one example you learn. Though
Yes,
fructose is given, I want you to learn only this first one, A1. B also you ignore, not required. Okay?
yes,
Yeah. Fructose mutarotation not required. Reactions of monosaccharides not required, beta. Nobody
yes,
is going to ask you theory wise. Glycosides, osazone form. Osazone formation is important in practicals, but anyhow you will see and learn there. Not at all in theory at all. Sometimes this glycosides can come for 3 marks better. What are
yes,
glycosides? These are formed when hemiacetal or hemiketal hydroxy group, learn the definition, of carbohydrate reacts with hydroxyl group of another carbohydrate. Till here, only this definition.
or
After that,
hemichaetal.
what you do is, after learning the definition, just name this better. What is the importance of glycosides? Okay,
Yes,
so like why do we need this glycoside? For examples, how do we use? Like glucovanillin, vanilla ice cream, what we use. Cardiac glycoside to treat heart diseases. Streptomycin, it is used as an antibiotic. Ubain, it inhibits sodium potassium ATPase and blocks the transport, like that. These are the therapeutic general uses of glycosides. So just learn the
yes,
definition, the first sentence, and give the uses. Okay.
yes,
So that is for glycosides, and nobody will ask you derivatives, not required. Anyhow, just simple idea. Maltose, sucrose, I gave you the table, no? From the table, whatever is given, you learn. All these are not required.
yes,
Mm, Polysaccharides. Polysaccharides, I said, if the sugar units are more than 10, monosaccharides
10.
more than 10 units, then you call them as poly. Homo means if it is made up of all same type
Or
of
the same type of unit.
monosaccharide units. Very good. Next example we have glucose. I said starch and glycogen. They are all made up of what?
Glucose.
Glucose. Very good. Heteropolysaccharides. So under this heteropolysaccharides we have glycosaminoglycans. Now we are moving to some important topics now. Homopolysaccharides, as you said, they are all made up of same type of monosaccharide units. For example, starch and glycogen. Starch, I said it is a plant sugar, plant storage form of sugar. Glycogen is animal storage form of sugar. Okay, so starch is a carbohydrate reserve of plants. Okay, example like glucosen means what? It is a polysaccharide chain of all glucose molecules. You call it as glucosen or glucan. Now starch, best example is rice. So suppose if you just wash the rice and try to just you want to cook it after 2 to 3 hours. How do you see that starch? It is light yellowish. That means what? The rice granule is getting dissolved to some extent, isn't it? So that means
Yes,
starch is made up of two polysaccharides, two components. One is water soluble and the other is water insoluble. Can we just soak the water in rice and can we eat? No. We need to cook
no,
it so that it becomes soft, gelatin. That means what? Starch is made up of amylose and also amylopectin.
pectin.
Amylose is 15 to 20%. Amylopectin, which is water insoluble, it contributes to 80 to 85%. So amylose is long unbranched chain. How many glucose units does amylose? See, always there will be a question, Anjali. What is the difference between amylose and amylopectin? You need to learn this homopolysaccharides carefully. Learn properly. Very easy only. So, amylose is water soluble, amylopectin is water insoluble. Amylose is made up of how many glucose units?
What is this 15-20% and 88%?
That means if you consider starch as 100%, starch
Hmm.
it has two components. One is amylose and
Okay.
one
Amylose and amylopectin. Okay.
uh, 20 plus 80, how much better? 100.
Yes, ma'am.
Like that. Okay. Today you are little dull, feeling
No, ma'am.
sleepy. Uh,
No, ma'am.
I'm finding like not as active as yesterday, day before yesterday. Okay, fine, no problem. See, amylose, it is made up of how many glucose units?
200 to 1000.
Yeah, 200 to 1000 D glucose. Okay, held by, I will show these linkages, alpha 1,4
Yes.
glycosidic linkages. Okay, then amylopectin, it also has linkages. Okay, let us see, otherwise you will not understand.
Ma'am, I had a doubt,
Ha,
not regarding this.
ha.
Uh, Today we had biochem lab, ma'am, and we did urine test. So
What
in
did you
that
do?
urine, ma'am.
Acha, urine. Okay, what did you
Ma'am,
examine?
so, we did first we did the presence of urea test, and then we did the presence of uric acid test,
Mm-hmm.
and then creatinine, ma'am.
Acha, acha.
Ma'am, in the if I mean this is to indicate the presence of all these components, organic components, right?
Yeah.
So how do you know that a person has the creatinine problem? I mean by these tests can you determine that? Uh,
Of course, urea levels actually we have to check in the blood, but based on different color changes, you should have noticed, no, Anjali?
Yeah, ma'am. I mean, does it, for example, for the creatinine, it got orange, ma'am.
Huh.
They asked us to take one test tube, they asked us to take the urine,
Mm.
and one test tube, they asked us to take distilled water, and
Mm.
then they asked us to add sodium carbonate, and no, is it NaOH, ma'am? And then they asked us for picric acid, saturated picric acid.
Acha.
And then the one containing urine turned orange.
That shows the presence of creatinine, right?
Yes, ma'am.
Mm.
So, I mean, if the color intensifies or something, do you know that? I mean, it's more
Creatinine
than.
levels will be more if it is intensifying. If it is light orange, then little less creatine. But anyhow, it's only just to check the presence, but to determine the exact values, we need to take blood sample.
But why are we determine? I mean, we know that urine already has all of these components, right? So, what is the use of these tests?
This test will determine whether the kidneys are functioning normal or not. These
Okay.
are renal function tests. When the person is suffering with renal failure, you will see all these parameters will increase. Sometimes
Yes.
patient may go into coma also. That's the reason we do
Yes.
this. Okay.
So if someone says someone has a creatinine problem, it means that if does it mean that it's overproduction of creatinine and over excretion or underproduction?
It indicates like creatinine is not being metabolized by kidney. Though it
Okay.
is produced normally only, it is not
Yes.
reabsorbed by the tubules of kidney. It shows renal failure. Okay.
Ma'am,
And
there
it
are
can
two
be
that
overproduction also, like in case of muscle disorders.
okay.
There will be excess production of creatinine.
Ma'am, what is the difference between creatine and creatinine?
Creatine is a like precursor from where creatinine is produced. Creatine will be converted to creatinine. That you will
Okay.
learn when we do protein metabolism. Okay.
Okay.
Got it? Okay.
Yes.
Now see, this is we were in amylose and amylopectin. So chemical structure of amylose. What is it made up of? I said 200 to
200.
1000
1000
glucose units.
glucose units.
Uh, and the linkages are alpha 1,4. Let me show you. What are these alpha 1,4? This is one, you are finding the cyclic form of glucose molecule only, not in the straight chain, but you have drawn in a form of a hexagon. Then you are finding there is one more glucose. Is there any linkage between them? Any bond? Where-
Yes, ma'am.
Yes. So the straight bond is called, like it is between first carbon and fourth carbon of two adjacent glucose molecules, isn't it? So you call
Yes,
this
ma'am.
linkage as alpha 1,4 glycosidic linkage. Got it? So
Yes, ma'am.
alpha amylose will have all these linkages only. If it has 1,000 glucose units, between these 1,000 glucose units, you have these linkages. Tell me what are these?
One, ma'am, for amylose, it is one for alpha linkage. It has 200
Alpha
to 1000
1,4 glycosidic linkage. You should say.
glycosidic linkage.
Hmm. For
Ma'am, for amylopectin, it is 1,6
1 6
alpha
and
glycoside.
1 4. I'll show you both.
1,4 also. One
Yeah.
six and 1,4.
Yeah. See, this is amylopectin, Anjali.
Yes,
For
ma'am.
adjacent glucose molecules, you have 1,4 linkages. Branching places, you see 3
1,6.
carbon and
Yes.
6 carbon. So it has
Yes.
both. Okay, straight chain will have 1,4 linkages. At branching points, it has 1 6
Chain is
linkages.
just like amylose. When it is, I mean, branching, then this one.
Hmm. That is the only difference. You have branching in amylopectin, whereas we have straight chains in alpha amylose. That's the reason there you have only 1,4 linkages. Here 1,4 and 1,6 both are there. Okay. Then. So that is all about this amylose and amylopectin, water soluble, water insoluble, how much is the composition and everything. So what is the enzyme which is working on this starch?
Amylase.
Yeah, amylase. Okay, then we have dextrins. These are breakdown products of starch. Okay, so inulin, you were telling, no, I asked you, give me one example. You said, yeah, inulin. It's a polymer of
Yes.
fructose, fructosen. Like glucosen we use there, here we use the fructosen. Okay. So basically inulin is not utilized by kidneys. It is excreted in the urine. Suppose if you want to check renal function test, if you are giving 1 milligrams in the blood, same 1 milligram should be excreted in urine. Then it shows kidneys are functioning normally. That's what
Okay,
is called
ma'am, what if it is absorbed? I mean,
observed in the nephron. But inulin
yes.
is not observed. Inulin is nowhere observed. Hence, we can use inulin to assess renal function.
Ma'am, so if, uh, I mean, how do you know that it's not functioning properly?
It will be retained in the blood only. It will not be seen in urine.
Okay. Yes.
That shows like it's not functioning. Okay. The next
Yes.
we have glycogen. What is as the difference? I said starch we find in plants and glycogen we find in animals. And
Animal
if you see the structure, it is much more complicated. There you had only 1,000, but here it has 25,000. See,
25,000.
it has straight chains, it has branched chains. Look at this picture. This is all glycogen. Straight, what is the linkage you have when it is straight?
Uh, Glycoside.
Alpha 1,4. 1,4.
Alpha. Okay. Alpha one.
If it is
So
branching,
1,6.
very good. So here, instead of drawing complete formula, all boxes are given. So straight
Yes.
boxes, between them line is given, nothing but alpha 1,4 linkage.
1,4 link.
Branching 1,6 linkage. Okay, beta? So
Yes.
that is about glycogen. Where do we find cellulose? I said plant
Plants.
fiber, what is there, no? And
Yes, ma'am.
see, when you look at the structure of cellulose, instead of alpha 1,4 linkages, it has beta. See, look like
Beta.
this. Beta, is it straight line or crisscross?
No, ma'am. It is a cross line.
So such we don't have an enzyme which can digest, which can break this bond.
Cellulose.
Hence, it is not digested in our body. Okay, so that is cellulose, which is constituting fiber. Read better; it's very easy.
Yes.
Heteropolysaccharides are very, very important. Hetero in the sense,
Ma'am, what if, I mean, how does it, for example, we eat leaves, no, ma'am? I mean, some some leaves. So
raw
how
things,
does,
raw things, whatever we eat like carrot, beetroot and all, that is rich in fiber.
yes, ma'am. So the I mean, there is cellulose in that.
That is made up of cellulose.
So how does it get digested?
It is not getting digested. It will get excreted in the fecal matter.
Okay,
But
so that is how the fiber is formed.
it makes like excretion easy.
Okay, ma'am.
Constipation will be prevented.
Yes, ma'am. Got it.
Okay. Ah. So heteropolysaccharides, heteroglycan, like I said, they are made up of different sugar units, not like glucan or fructosan.
Yes, ma'am.
Under this, no, you have a table which you have to study. I'll show you. Heteropolysaccharides. This is the most important topic. See, this table you have to learn. Pakka question this is. Heteropolysaccharides also called as glycosaminoglycans. You have 5 examples here: hyaluronic, chondroitin, heparin, dermaton, and keratin sulfates. You need not learn this structure. See here, look at the screen continuously, Anjali. Hyaluronic acid, what is it made up of? Read and tell me.
D-gluconic acid and
D-glucuronic acid and N-acetyl
N-acetyl
glucosamine.
glucosamine.
What type of bond you are finding? Is it alpha or beta?
It is beta.
Beta. Linkage 1,3.
Yes, ma'am.
Isn't it? So, for
Yes, ma'am.
example, now hyaluronic acid has come under glycosaminoglycan. You will write its composition by looking at that picture. You need not
Gluconic acid.
learn these structures at all. It's only for your information. Okay.
Okay, ma'am.
Then, where is it present? Hyaluronic acid in connective tissue,
Connective tissue, synovial fluid, and vitreous.
vitreous humor in the eyeball. What is the function?
Serves as a
Lubricant.
lubricant, shock absorber, and wound healing.
Promotes wound healing. Similarly, you have remaining 4 also. Just look at the structure, remember the composition, where is it present, and what are its functions.
Yes,
Okay.
ma'am.
This is the most important topic among all, which usually comes from carbohydrates chapter. Okay.
Yes, ma'am.
Glycoproteins. What is the meaning? Glucose in combination, like
Of
carbohydrates in
protein.
combination with proteins. You call them as glycoproteins. Okay. Few examples are given, like collagen is a glycoprotein. Okay, it gives structure to the cell. Hydrolases, proteases, glycosidases, these are enzymes. Like that, few examples are given which are glycoproteins. Immunoglobulins are glycoproteins which kill the microorganisms. Okay, like that, few examples you can learn. So that is all about this. Check what are the possible questions, essays, shorts, try to fill up the blanks. Okay, and check
Yes,
these multiple choice questions also.
ma'am. I mean, after every chapter, there are these questions, no, ma'am. So if I solve these questions, is it enough?
More than enough.
Okay. So if I just give a reading of the chapter
Huh.
and then try to uh, answer all of these questions, enough.
More than enough. And you will be submitting these answers to me without seeing.
Yes,
Okay.
yes,
Write this test in the